Welcome to today's special seminar of the Julian Wrigley Global Futures Laboratory. It's a great pleasure for me to introduce Amy Maas from the Bermuda Institute of Ocean Sciences as our today's speaker. Her talk title you already have seen is on improving estimates of zooplankton contributions to biogeochemical cycling. Amy is a comparative physiologist and a biological oceanographer. Her research addresses fundamental questions about how the environment influences the distribution and biology of marine invertebrates. Based on understanding these cycles, she works on identifying how climate change will impact biogeochemical cycling. Her current research projects include studying theropod distribution and seasonality at the BATS site. BATS is the Bermuda Atlantic time series. There's also a HOTS site, which is in the Pacific, the Hawaiian Ocean time series. She also explores, her work includes explorations of circadian rhythms and vertical migration in open water zooplankton to better understand how environmental changes impact carbon flux and comparative transcriptomics of multiple plankton species. All of that, of course, has to do with our changing planet, the changing environment, changing oceans. What does that mean for ecosystems and how ecosystems influence the larger ocean environment, including the so-called biological pump of carbon? And Amy will explain all that much more in detail than I can do that as an outsider to marine biology. A little bit about her background. Before joining BIOS in 2015, Amy was jointly appointed at the University of Connecticut Marine Science and Technology Center. There she was an assistant research scientist. Between 2011 and 2015, she was a postdoctoral scholar investigator at the Woods Hole Oceanographic Institution. And there she worked in the biology department studying the sensitivity of marine ecosystems to ocean acidification. Amy received her PhD from the University of Rhode Island in 2011. With that, I would like to hand over to Amy.
Thanks, Peter. I really appreciate that. Hi, everyone. I am going to make sure this all works. And good, we're advancing. So this is what Peter basically just said. And I think one of the things that I wanted to say to add to that, just sort of how I got to where I am at BIOS, is that I started out, I'm from Ohio, at a liberal arts college, just studying biology. I moved into biological sciences department at a place that had an oceanography school, and so had that sort of marine experience, and then went to Hui, which is very strongly oceanographic. So one of the things I'm pretty excited about with ASU is actually being able to step back maybe a little bit more to that liberal arts perspective in that broader context. And the work that I do is all over the world. So these stars are sites where I've published papers, worked on things, and so you can see there's a relatively large geographic spread. And my topics have shifted and merged and gone back to each other. As Peter said, I started in ocean acidification, but with a heavy emphasis on where organisms are and how their physiology is affected by that distribution. I've dabbled a bit with collaborators in biomechanics and how these other things relate to swimming capabilities, some work on biodiversity. But more recently, my work has been very heavily focused on biogeochemistry, and in particular, the flux of carbon and nitrogen in through the ocean systems. But I don't feel like that actually encapsulates why I do what I do, right? So I think it's important to take a step back. Again, as someone who grew up in Ohio and had no real understanding of what the ocean was for, I think that the takeaway message I got after years of working in the ocean environment is this understanding that we really are truly connected to this ocean system, and that it is an important shared resource. It's a global environment that is really large, but it's also an environment that provides a really important ecosystem function for humans. So aside from providing food and oxygen, it balances a number of biogeochemical budgets, heat budgets, and really influences climate. And so as human activities change the ocean, there are feedbacks that influence humans. And the particular context that I'm going to be talking about today is the flux of atmospheric carbon into the ocean system. So this is this passive relationship between the ocean sink and the atmospheric sink of carbon. And for those of you who are unfamiliar with the way it works, we have atmospheric CO2 that is just moving about the globe throughout the atmosphere, exchanging with all of the sources and terrestrial environments. When it comes into contact with the ocean, the CO2 passively dissolves, is taken up by the primary producers of the ocean system. These are our phytoplankton. Okay, and unlike terrestrial systems, many of these are quite small. And here, they only have a thin area where they interact with light. So photosynthesis can only occur in the upper water column in what we call this euphotic zone, or EZ. And so they're at the surface of the ocean. Remember, that's like a very thin skin on top of the huge depths of the ocean. Much of the ocean has no light in it. And so the transport of the carbon out of that surface area down into depth is really critical. As it moves through the ocean system, there's a number of different ecological processes that occur. Some of them are mediated by biology. We have grazing and the production of waste that can then sink out of the water column or be mixed out of the surface area. This is done by small, large, and charismatic organisms as well. There's also physical transport by the processes of mixing wind energy, as well as other just natural gyre systems. And as this carbon moves through the system into what we call the twilight zone or the midwater, it moves away from where photosynthesis can occur. So it starts to be the only source of food for everything in the midwater down to the ocean floor. And because of that, it starts to get worked. It gets eaten. Things are coming in, encountering these particles and taking away that carbon, making it part of their biomass and preventing it from getting deeper into the ocean. That is, this loss product is called attenuation of flux, right? And after a certain point, that carbon reaches the seabed. It's there, it's sequestered, it's in the midwater, it's in the sediments. There's not really a good chance that it's going to come back up into the atmosphere. And so the importance of this is that it controls this global carbon cycle. But also remember that that really tiny thin layer is providing all the most of, it's not all, there's other small places, steep vents, et cetera. It provides a lot of the primary productivity, a lot of the carbon for the midwater and most of the benthic biota. Because when that carbon gets fixed, all the other micronutrients and macronutrients, such as nitrogen, phosphorus, as well as iron, are part of that biological material. There's links between that sinking flux and the drawdown of nutrients in the surface water. And so the nutrient concentrations in the surface water are very highly tied to this flux of carbon. Now, the piece that's really important, and the reason that there's been a lot of focus on this right now, is that originally the oceans were actually a little bit of a flux out of carbon, a positive flux into the atmosphere. But with anthropogenic activities, the ocean is currently acting a bit like a sponge. It's taking up 30% of the anthropogenetically produced CO2, okay? So that's 30% of the extra CO2 we are producing is not going into the atmosphere, is not contributing to global warming because it is being drawn down into the ocean. Understanding the mechanisms by which that drawdown occurs, the mechanisms by which it can flip, there can be a tipping point, it can switch. Those are the reasons that we're really drawn to understand how this flux system works. And the particular components that I've been working on have been related to the zooplankton. So that was my center slide that shows some of the diversity of zooplankton. Now, zooplankton, in the definition I'm using right now, are animal plankton. Specifically, I'm referring to multicellular or metazoan animals. And plankton is organisms who don't have enough swimming power to swim against the major currents. That's not to say they can't swim. They swim up and down all the time, but their advection through the system and their connection to different places is really strongly dictated by the big gyres. They get caught in eddies, they travel with the water. So the physics of the ocean are constraining where their habitat is controlled. And this is pretty much any metazoan phyla that can exists in the plankton. I've mostly studied, here on the left, you can see the pteropods. These are shelled mollusks. They have calcium carbonate shells. Instead of having a foot like a snail, they flap with little wings in the water, absolutely gorgeous. And the reason I study them is because of that calcium carbonate shell, which has an added flux component that it is bringing calcium carbonate, which is affected by ocean acidification. The other group that I work strongly on are the copepods. And the reason that we study the copepods is that they are the most numerous, abundant, highest biomass, and most diverse of the metazoan organisms. These are the insects of the ocean, like they're pancrustacea, they're actually related. They are the dominant source of biomass in this trophic level is the copepods. And so I mostly work on those two for the specific reason that they're really good indicators of what's going on in the ocean system in the context of climate change. And to get into the minutia or the details to try to bring you guys into where the science is that I'm actually working on. If you look at the role that the zooplankton play in this biological pump, this movement of carbon from the surface, drawing it away and down to depth, the zooplankton have like two big roles. One of the roles is that they moderate the sinking stuff, passive flux. All right, this is stuff that's produced in the surface or even in the midwater that just naturally sinks down to depth. So we have that carbon that's being fixed by the net primary productivity. So this is all the plant life. They're taking that carbon in, they're making biomass, and that's being consumed by the zooplankton. And the zooplankton are busy swimming about doing their thing. But some of the carbon that they ingest is excreted as fecal pellets, particulate organic carbon. And that sinks out of this euphotic zone and down into the twilight zone. Here, midwater organisms repackage, eat that carbon, and they transform it. Some of it ends up in smaller particles, some of it ends up in larger particles. And those processes change the rapidity with which the flux moves through the water system, changes the ability of that flux to move away from the surface. And remember, the faster that that flux moves away from the surface, the more likely that it won't get mixed back up into the atmosphere and become a problem for global heating purposes. So we want, in our very selfish human way, we want that flux to be sequestered deeper down. And the easier way for things to get sequestered is to be packed in one of these particulate organic particles. The other role that zooplankton play is a little harder to trace because the products are more invisible. On a daily cycle, zooplankton make the largest migration on Earth. So at night, they're up in the water column consuming net primary productivity. But then when the sun comes up, all their predators can see them. And it's not like the terrestrial system where you can just be like, oh, I'm going to go hide in a cave, or I'm going to climb up this tree. Like there's no place to hide nearby. So organisms can either be small and transparent, or they have to make this massive trek deep down into the water column where they can hide from their visual predators at depth. So this diode vertical migration is this trade-off between energy acquisition, which is much easier to do in the surface water, and mortality avoidance, which is much easier to do deeper in the water column in the dark depths. So this diode migration occurs every day. Not all species do it, but those that do, they consume most of their food in the surface water. And then when they swim to depth, they're still excreting, they're still producing waste products. So it's this shunt direct from surface down to mesophotic level. So usually they're about 300 meters to 500 meters. So it's not very likely that that carbon is going to come back up to the surface. And the kind of carbon that they produce is respiratory carbon, CO2 that they breathe out. And then they do bring some of that fecal pellets down to depth. We call this gut flux. It's a nice way of saying they're pooping. And then dissolved organic matter. So they're excreting dissolved pee at depth. And these processes not only move carbon through the system, but they really strongly dictate what is available to the food web in the midwater, because they're bringing things that are high quality down into areas that usually only are just waiting for manna from heaven, the little particulate rain of things from above. So there, this teleconnection between the two layers of the ocean system that sort of bypass some of the sinking properties. So zooplankton play this role in the biological pump. And in the context of climate change, I'm trying to understand how variation in environmental conditions change these behaviors and these physiology. Because we know in the broader context that as human activities change these environmental conditions, there's going to be changes in biodiversity, winners and losers in systems. They're going to be outcompeted due to changing environmental conditions. It can also change ecosystem function, the way that carbon flows through systems and how food webs function change as the physiology or the organisms change. And because we know that this system is already responsible for a huge amount of additional CO2 drawdown, there's potential for climate feedbacks, right? We could have acceleration of our climate change experience if this drawdown is changed. So that's sort of the context for the work that I'm going forward. And the way that I do it is I use this pairing of very old classic tools, because they work, with some very new modern tools and data pipelines. And one of the biggest tools that we use is we have this massive net. It's called a MOC-NES, which is multiple opening and closing net environmental sensing system, which really what it is it's just a big set of 9 stacked nets. And you set them open, closed, and you send it deep, 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 deep down into the water. And then you start to pull it up, and then you close it, and the next net opens, and you close it, and the next net opens, and you close it, and the next net opens. You get the idea. And at each of these depths, I have measurements of what the environment is like. So it comes back on board. Each of these contains organisms from different environments in the water column, paired to oxygen, light, temperature, salinity, you name it. And in the good old days, we used to just maybe count them, weigh them, and then figure out things from there. Nowadays, what we're starting to do is use tools like this, optical measurement systems. This is a ZooScan, which is really just a very expensive waterproof scanner. It produces lovely images, however, silhouette images of the organisms that we're studying. And we put this into a assisted machine learning program that allows us to identify the different taxa that we're looking at. And it also gives us a whole bunch of morphological metrics, like how long, how wide. I can use this to estimate how big an animal is. By that, I mean biovolume. And from there, we can also say some things about their shape and size that relates to their role in an ecosystem. Paired with this, I do a whole bunch of physiology experiments. So these are little respiration chambers that I've designed. Inside of each of these chambers is a small zooplankton, and I'm allowing it to breathe under different environmental conditions, whether that is temperature. I've tortured them with every possible thing you can think of: oxygen, CO2 level. We've even done ammonia toxicity. So looking at these organisms' response, I look at their oxygen consumption, their respiration rate, and I also measure things like their excretion rate. So we'll take water from these chambers, measure how much dissolved organic carbon they produce over time, how much particulate organic carbon over time. I also do a number of more detailed physiology related to transcriptomics and proteomics, but I digress. From these sorts of data sets, what I can get is I can rely on certain relationships that have been shown to exist throughout much of biology. This is called the Almetric scaling curve. And what we know from our studies of organisms, all types of organisms, is that there's a relationship between the size of an organism and how much energy it requires. Now, there is a fair amount of spread across this line, but the relationship itself is solid. So this is called a scaling exponent. People disagree on the scaling exponent. That's not the purpose of this talk. The important part is, if you can estimate how many animals you have and approximately what size they are, you can use these equations to calculate the metabolism of the community. And from there, you can say something really powerful about how this carbon is moving through systems. And it's one of the better ways we have it, because we can't well measure the changes in oxygen or CO2 in the background of the ocean system. It happens too fast. The signal noise is not good enough. So you have to be able to characterize it using these calculations. Unfortunately, if we take global measurements where we've done both the migrant biomass and the respiratory flux, you see that the relationship is pretty inconclusive. So if we know the migrant biomass and we apply these relationships, we still get very different calculations of respiratory flux. And so from this, we know that this is one of the uncertainty areas in our understanding of the flow of carbon through the system. It's been decided amongst the community that this is a point of growth, that the biogeochemistry needs to do a better understanding why migrant biomass and respiratory flux relationships haven't been nailed. And we really think that we're missing some important things. Figuring out which things are important and which things are just interesting is what I've been trying to do for the past couple of years. And so that's mostly what I've been working on. I've been predominantly focusing on this Diode vertical migration active flux box. And so that's what I'm going to talk about today. Some of the research directions I've been doing to try to improve our modeling of this movement of carbon, and see if there are some things that we've been overlooking that we should possibly consider because they are contributing to that 40%. Let's get that down to like let's get to 10%. That would be great. And what we've been doing is, again, pairing these old mock nest toes with these image analyses. And what we can do with these image analyses is we can start basically making histograms, counts of how many individuals you have in a particular size bin during different times. So we remember the animals, sorry, that was my tea. At night, all the animals are up in the surface. So this histogram relates to this depth strata in the water column. So this is the well-lit epipelagic zone. And at night, you can see there's a lot of animals there. In the day, many of them disappear. And if you track down here, you see that they're mostly showing up down in these depths, which is between down to 500 meters in the water column, okay? And so with these histograms, we can do zooplankton particle size class magic. Basically, I'm just taking nighttime versus daytime. And you can see that these small migrators are moving to these depths, and these large migrators are moving to even deeper in the water column. And from there, what I can do is I can calculate what the physiology of this size and abundance of organism is and see where the carbon is actually going in the water column. And if you remember, if we go back to that human-centric idea, we want the carbon to be deeper. So where and which organisms are important for moving that carbon deep in the water column? And how does that track seasonally over the course of the BATS time series where I'm at? Or how does that track along gradients in the ocean system so that we understand where do organisms do a better job of moving this carbon? I can also look at the animals that don't move and assume that those animals, particularly in the midwater, are getting their carbon from intersecting particles. And so these are the guys who are reducing the efficiency of flux. So seeing where they show up can also be really important. Another piece that we've tried to focus in on that's been a point of contention within the community is how important is it to study different kinds of animals? So on this side, I have a community that is mostly these crunchy copepods that I said were super important. Crustacean zooplankton. They have different feeding patterns, body composition, and life history than this type of organism over here on the right. We see salps. Now, salps are gelatinous tunicates. They're basically like vacuum cleaners hoovering up the ocean. They're just a mouth. And they're really efficient grazers, which means they're also really efficient poopers, right? So they move carbon in a very different way. They have lower mortality, however, and their respiration rate is lower. So if you have a community that is predominantly made up of mesozooplankton, you may see that they graze less, but they produce more carbon flux as they transport. Salps graze a lot, but most of that is coming out as fecal pellets, which is this yellow bar. So trying to basically take the equations that relate to these different processes, plugging them into community composition and see how important does taxonomy matter, particularly because we are predominantly in many ocean systems dominated by these crunchy crustaceans. Salps show up sometimes, but sometimes they're really important. Using this kind of data set, we realized that there's vast changes in the analysis of how much metabolic activity organisms produce if we use this image-based data set rather than an average sized individual. And if you think about that, allometric relationships say that you're going to have exponentially higher numbers of small animals than larger animals. So if you take the average size of an animal, you take the biomass, divide by the number of bugs you count, that average size bug is not really going to be representative of the community because you're skewed exponentially towards smaller individuals. The other thing about allometric relationships is that smaller individuals have a faster metabolic rate per biomass. So the smaller bugs breathe faster anyways. So with those two things added, it's really having the individual size of each particle, each zooplankton, gives you a substantially higher metabolic activity. So that's, this is data from Bermuda Atlantic time series, 3 cruises that we did, one in 3 different time points. If I use the average size bugs, you get a pretty small flux. You get substantially orders of magnitude higher flux if you use image-based estimates of metabolism. Now, the community here at BATS is mostly copepods. So adding taxonomy doesn't make a huge difference here. And we never really ran into something like a salp bloom here. First glimpse at other data sets, that does matter elsewhere. So using that kind of data, we're starting to see places where we think that size is going to be a really important trait. And in conversations with modelers, there's ways of implementing size into our biogeochemical models to already start improving our estimates of how these zooplankton move things through ecosystems. I think, however, there's some other things that we're overlooking. And one of the big things is the physiology of migration itself. So this is kind of a visual picture for you, for those of you who are not used to thinking about this migration. Again, we've got these organisms at night, they're at the surface, they're consuming food. Then during the day, they swim like hundreds of meters, like think 43 marathons in body length down to depth during the day. And then they come all the way back every night. And so do we think that that potentially might influence their physiology? Probably. Do we think that there's potential changes in their metabolism over these timescales? Probably. There are also differences in the environment between these two regions. At depth, it tends to be colder. There's often less oxygen depending upon where you are in the ocean system, and there's a lot less food. So when we think about these things, as we are currently modeling, we assume that their metabolism is more or less the same over 24 hours, and that the differences between these two time points only has to do with temperature effects. So if we're trying to really nail some of that biogeochemistry, it's important to consider whether we're thinking through the when and the where things are happening. And so that's what I'm going to talk about next. We have demonstrated that there truly are circadian rhythms in the respiration of zooplankton. So what we do is we take animals, we keep them in a respiration chamber for a period of time. And this is a measurement of the respiration rate. Now, the first thing you see is that the metabolism is declining over time. They're in a chamber with filtered seawater. They're starving. They're not happy with me. They're running out of energy. Despite that decline in metabolism, we see peaks in metabolism every 12 hours. And those are indicative of a circadian rhythm. This was done all in the dark, so it is a natural and trained endemic rhythm. And what's interesting is that they peak at dawn with the lowest point at night, which is somewhat counterintuitive if we think of them being very active at night. Based on these, with a single individual, the changes in respiratory flux could be between 6 to 24% for the species that we studied here in the Sargasso Sea. Another thing that we documented was the production of fecal pellets in the early morning at depth. Typically, when we make our estimates, biogeochemical estimates of zooplankton contributions, mostly the community just sticks with carbon dioxide because it's the thing that's been measured the most. We're starting to add dissolved organic carbon, but many researchers, even today, don't include fecal pellet production or mortality at depth. And it looks like those things are going to be important. We did an experiment where we caught animals 4 points during the day in their natural habitat, twice in the surface, once when they first came up, once in the middle of the night, once when they first went down to depth, and once later in the afternoon. And we're finding that even 6 hours after they've left the surface water, they're still producing fecal pellets. So this is carbon that is being moved, shunted down to depth. And this is the pictures of surface versus deep. Okay, maybe there's less in their guts, but there's still stuff coming out. And so incorporating that into our estimates is important. My screen just did something weird. Can you guys still see it?
Yes, I can see it.
Okay, I can't, which makes it a little bit complicated. Give me a second. What do you see?
I see the header dissolved organic carbon experiments. And now it's switching around. Now you have.
Okay, let me get to where I'm supposed to be. Roger, I am now where I need to be. Okay, couldn't see it. That was tricky. And we're back. Okay. The other thing that's important is that if we investigate this slightly more complexly, if we look at the gene expression and the protein expression of these organisms over the points of the day, we see that the way that this kind of graph, if you've never seen this before, is that these are different genes that are being turned on or off. If they're differentially expressed in a positive, they're yellow. If they're negative, they're purple. The colors are the times. What this is showing me is that there's clusters in the expression profiles of organisms based on time of day, and that there's a lot of genes being turned on when they're at depth. The protein says similar things. So there's a lot of activity happening at depth, and we canonically think of them being down at depth and just piecing out and zenning out. But what we seem to be seeing is that there's a lot of translation, transcription, and metabolic activities that are happening at depth. What's important about that is that if they're still doing different physiologies at different times of day, their waste might be different. So the big picture that we have is that when we measure them, they seem to be doing a lot of tuning to try to keep their metabolism running throughout the course of the day. Some things like enzyme levels that we have been using as a community to compare between day and night have different profiles, circadian profiles, which suggests that those methods are not actually appropriate for studying differences between the day and the night. And because these different processes are happening at different times, waste products likely vary over the course of the day. If you're metabolizing things differently at different places, if you're doing protein assembly at different times, your nitrogenous waste is going to be happening at different times of day. What's critical about that is that if you know when it's happening, that influences where it's happening. Because if it's happening at night, it's at the surface. But if it's happening during the day, it's at depth. And there's a lot of basic questions that are still unresolved here. How much waste do these organisms produce? Is it different by different taxa? What exactly is it? I mean, carbon, nitrogen, right? But what? Does it matter? That's the final question. Is there just like a definitive proof that this is something we should be wasting our time and NSF dollars on? So to address that sort of question, I'm going to talk to you about a project that I did as part of this broader Bioscope collaboration. And this was really these dissolved organic carbon experiments. And the idea was we would catch live zooplankton of the species that's most abundant out here. We'd wash them 1 million times and put about 60 of them in a cubitainer with a bunch of filtered seawater. And then at the end of the incubation where these organisms are excreting dissolved organic carbon, we remove the zooplankton, and then we'd measure what's in the water: metabolites, dissolved organic carbon, and dissolved free amino acids. We would then take a portion of this water and we do what we were calling remineralization experiments. We would inoculate this water with seawater, natural seawater from different depths in the water column, and see how the organisms from the twilight zone responded to this pulse of dissolved organic carbon from zooplankton. So we did those over the period of 14 days, and we called that remineralization. And so first steps, dissolved organic carbon. How much do they produce? So they produce an appreciable amount of dissolved organic carbon over the about 12 hours that we were measuring them. If you put this in the context of the circadian rhythm studies, most of this production would have been done when these organisms are at depth. Some of it would have been right before they're about to swim to depth, because we always catch them at night. But most of this is the kind of metabolites and dissolved organic carbon that would be produced while they are in the deep. Of that, a lot of it was dissolved free amino acids. And the reason that's important is a lot of the carbon that's in the midwater of the ocean is actually not yummy stuff. It's kind of like the dregs of things. Think like Victorian London. These are the stuff that managed to make it into the gutters. It's like the rag pickers and the bone sellers. You know, it's not the nicest carbon. And the way that we measure that, one of the ways that we measure that is we look at certain compounds and we see how what the diagenetic state is. So is it yummy carbon with this low diagenetic state, or is it sort of the worked over recalcitrant carbon that's harder to metabolize, harder to work over? And so we see that the kind of compounds that these organisms are producing is the yummy stuff. So this is rich labile carbon that is delicious to eat. To get a little more specific, because we can, we're collaborating with Liz Kaczynski's group at HUI, and so she does high resolution DOMS, she does metabolite analysis, and so she's taking these dissolved metabolites and helping us identify what they are. This is how it works. They never would let me touch that machine, not my job, but they do give me the cool data. And what the data shows me is that some of the things that these organisms are producing are very interesting compounds, such as vitamins and nucleosides. Now, these are things that if you measure in the water column, you're never going to pull up a vitamin, because as soon as a vitamin is produced in the water column, someone's going to claim it. So we're seeing these exchanges that are happening so fast that they don't build up in the water. So we don't have a measure of these things being produced in the water column. As soon as vitamin B10 shows up, something's going to take it out of the water column, because it is a rich labile thing. So this is a pathway that hasn't been measured for for kind of important metabolites that we didn't have a sense of, or at least we think they're important. Can we actually prove that they're important? So that's the remineralization experiment. We're taking the bacteria, putting them into the dissolved organic carbon and seeing how they respond. So these are 14-day experiments. What these are is DAPI counts. So we've labeled the bacteria fluorescently and we're counting cells, okay? Now, I should have mentioned, there's 3 treatments in here. There's the control, which is, right, nothing happened to the water. There's a carryover treatment where I was worried about how much dissolved organic carbon was coming through from the water rinses, and I'm calling that the carryover control. And then the copepod one is the actual experimental. So we have basically a control, a blank, and a copepod. And you can see that bacteria respond significantly to the presence of the dissolved organic carbon that's in this water. But you also see sort of the senescence, and over time that community tanks. The amount of dissolved organic carbon, again, we showed previously that there is the production of dissolved organic carbon. But by the time that bacterial peak occurs, they've chowed down all of that DOC basically back to background level. So everything that was yummy and delicious, they've removed, and it's back down to this recalcitrant normal level of DOC for the rest of the experiment. This again is that DFAA, that highly delicious, yummy carbon, dissolved free amino acids, and it's gone by day 5. Yes, we should have done a higher temporal resolution. I think I need to write another proposal for that one. And if we investigate this by looking at the community that's there, what we see here is this is the normal background community within the Sargasso Sea. So you have a bunch of what we call these SAR for Sargasso, clades. These are oligotrophic bacteria. They are good at eating recalcitrant stuff, but there's also a fair amount of diversity in the water column. Now, we do have a bottle effect that occurs. So this is our control and carryover. You see that both the control and carryover look substantially different from the initial, but they are also substantially different from the copepod treatment. And so statistically, we're seeing a move. Another way to look at it in a statistical way is that at time 0, this is a very oligotrophic ocean bacterial community. Over time, you get these Alteromonas, which they're a weed species. They do really well. They outcompete and consume other bacteria. But the copepod treatment is enriched with these groups called Flavobacteriales, Rhodobacter, and Pseudoalteromonidea, Pseudoalteromonas. There's a couple of specific lineages that we're especially seeing show up in this DOC community water. I wanted to make one more point here. And these are organisms that exist in the midwater. They're copiotrophic bacteria. They are down there and they're waiting for these opportunities to bloom, right? So this in some ways is increasing the niche diversity of that mesopelagic zone, that there are opportunities as these organisms migrate up and down to increase the complexity of the niche if they happen to be intersecting with the zooplankton who's exuding this cloud of DOC. This experiment doesn't quite capture even the DOC concentrations because I have these animals excreting inside of a 1 liter thing. The clouds that they would excrete would more likely be very close to their body and then they jump away. But because of the filtering and the way that it all happens, it's not entirely representative of what's going on there. We've talked about trying to label it fluorescently, but haven't come up with a good way of doing it. So the take-home messages from this whole sort of thing is that there are a lot of new tools and technologies, things like these optical systems where you can simultaneously get the taxonomy, the identity, the number, and the biomass of organisms. And these are starting to become automated systems. So there are systems that are going out there. We're working collaboratively to try to observe, literally observe with cameras, the ocean system. And as we start having more of these time series, there's going to be expansion in the use of these allometric relationships to try to understand the movement of carbon and nitrogen through systems. Physiologists then need to work with modelers to tell them the uncertainties and the gaps and to really fill in the information that is missing from different taxa. There's still, however, a lot of complexity in zooplankton behavior and physiology that isn't incorporated. We just make these large scale, back of the envelope, over 24 hours, divide by 12. And some of it seems to really matter. The circadian stuff does really seem to matter. And figuring out which things we can just say, oh yeah, that's cool, and which things have an appreciable change on our biogeochemical budgets is critical. finally, there's some basic information about excretion pathways that seem to be better parameterized, and we're moving a lot of that forward right now. And the really simplified message is that zooplankton are cool and you should love them, and their physiology is interesting. And even if you're from Ohio, their ecology affects your global biogeochemistry. And from there, I wanted to just transition into sort of the future directions, what's happening with me over the next 3 years. The data sets that we're working with right now are extensive. So we are producing similar optical data sets to what I showed for a number of different ocean regions, and trying to then establish basically the rules or parameters of the way that environments interact with these size and physiology relationships, as well as the depth at which organisms are moving. And so these are MOCNESS data sets from a bunch of different grants that help us get at both temporal and regional variability. We're also working on some technology development. So we have collaborators who are producing new instrumentation that can be chucked overside, and you can take your boat someplace else to do other work and then come back and recover it, that helps us really hone in on the depth at which these organisms are moving. On the left is a figure from a paper by Oman and Brockman in the California current system, and they have this toad system where during the night they're measuring the size of a copepod and versus the depth at which it's found. And they compared the nighttime versus the daytime. And you can see this really fine resolution that's based on the size of the organism. We want to be able to do that, but we want to be able to do that with an instrument that you can just leave and then come back. And so we're working on this project called Vampire that is hoping to help us get to this kind of data set. Aside just from size, there's a couple other what we're calling traits that are turning out to be really important for flux. Stay tuned. Watch Blanco Bercial's talk next week. That's more his thing. The importance is that, again, human-centric view, we want that carbon to be deeper, right? We want that carbon to be pulled away from our atmosphere. So where's the hot spot for organisms moving the carbon deeper? The other thing that I'm starting to focus a lot on is more on this sinking stuff, on this passive flux pathway. We have a proposal funded with Susanna Neuer of ASU looking at the role of zooplankton in producing these sinking aggregates. So that's ongoing right now. I have another pending proposal working on that. And some of the things that's important about that is we're realizing that we're going to have to apply different tools, because once particles are made, they're instantly moved around and repackaged and modified. And so some of the only ways you can try to trace them optically, or like microscope, visual, or using microbial analyses, but biogeochemistry is a really great tool. So looking at isotopes, amino acid specific isotopes in particular has been really helpful, as well as some other geochemical tracers like carbon and nitrogen ratios help us really understand the way that these particles are shuffling through the ecosystem as they move through trophic levels, they get consumed, repackaged, and broken apart, how that affects what's going on through the water column. And finally, this now is happening with ASU. What are the things I'm looking forward to? So currently, although I collaborate extensively with a bunch of students all over the world, I don't have students of my own. And I have a bunch of projects. So the things that I want to have going forward is I'm really interested in having graduate students. And I also have a number of projects that are really tractable to be worked on by undergraduates remotely. So I have ideas on how to move that forward. And that's sort of one of my big growth points that I'm really excited about. The other thing is that I work on a problem that can't actually be solved by science. We know the science of climate change. Right? We have uncertainty envelopes, it's true, but the really big problem that we have right now is getting political and societal motivation to move forward, to check on solutions, to make change. And to do that, you don't really need more basic research. And so one thing that I've gotten involved with is transdisciplinary work. And so I work with authors, artists, and we even have a new collaboration that's recently been funded working with a video game designer to try to bring the ocean to people who wouldn't have access to it otherwise. Because if you don't know something exists, it's really hard to love it. And if you don't love it, it's hard to work to change it, because people are going to have to give up things or change in order for this future that I can envision to unfold. And so what I'm really excited about is the greater opportunity to engage with people who are thinking about communication, thinking about policy, thinking about societal change and seeing how that comes forward. And with that, I have to thank my funding and my collaborators, which are extensive, and you for your attention. Nikki, I believe I'm ready for people.
Terrific. I'm Mickey Kittleson, Associate Dean for Faculty Success in the College of Global Futures, and I'm going to pass along questions to Amy. And so please feel free to enter your questions in the Q&A here online on our Zoom. And we have our first question in. When using nets and ZooScan to measure the zooplankton abundance, how do you design the sampling location to ensure the sampling results could be upscaled?
Yep. So we have right now, for example, we have 2 transects that are one along the east coast of the US, one along the west coast of the US. They were taken in 2 different years, but during the same month. And so we have all these environmental variables that are being compared across those things, but at the same time of year. And that lets us get at, with the oxygen layers, et cetera, et cetera. Those were all taken at the same depths so that we could cross compare. Here at BATS, we do seasonality, and we very distinctly, this is a human thing. We tend to try to ping environments based on depth, but the animals actually don't care about depth, and they're more interested in light and food availability. And so catching communities based on those parameters are more important. So each project that I mentioned is very targeted in picking times of years, regions, and depths to exactly get at what you're asking, upscaling. And the power is actually having broader geographic coverage and adding those all together to understand ocean systems, right? So using time series to get temporal, and then using these spatial stations to try to add that other dimension. So where we are right now is still really in the infancy to address the scale of things in the ocean environment. And I think the ocean observing systems that are coming online are what we're going to eventually need. One thing that people have been doing is adding things to floats to try to increase that coverage as well. So right now it's a patchwork. I would say we're still in the beginning. Did that answer your question?
I think it did, but they can add more to the chat if needed. The next question is, do the migrating organisms actually have to swim down active, or are they able to sink down passive?
Depends on the bugs. So my little pteropods, they are very negatively buoyant, like profoundly. They're heavy, they're little rocks. But that means they have to swim harder to get back up. A lot of the crustacean zooplankton are actually neutrally buoyant. And so it appears that they have to both swim down and up. The bioenergetics of how that happens is a little bit confusing, but we have some really interesting data that does suggest that this swimming up and down is evident even in some of the physical data. So they're actively moving up and down in the water column. And we see this in their gene expression as well. And when you first get them, when they come up to the surface, there's all this like myosin repair. So I think the same as after a big, long, heavy run, their muscles are needing some TLC.
We have plenty of time for other questions. So if anybody wants to add more questions in here, we have something in the chat here that says some. Okay.
Yep. So there are organisms that stay in the surface, these resident organisms, and actually there's residents throughout the water column. What we do know about their strategy, again, tune into Leo's talk next week, is that a lot of them work on being transparent. So they are clear, but there's trade-offs to that as well. The stronger of a swimmer you are, the more muscle you have, the less clear you are. So the ones on the surface tend to be smaller and more transparent. And there's like a sweet spot between size and transparency that means you have to move to depth. If you're big and dark, then you're a resident, but you're a resident at depth. And the ones in the surface, that they're living in the land of milk and honey, right? There's plenty of food up there. So they just have to stay small enough that they can outcompete the mortality piece.
Dave Gustin would love to hear more about your communication and informal education work.
Sure. So for a lot of this, I collaborate. We have a wonderful outreach coordinator. She's our K through Gray person here. Her name is Caitlin Noyes, and we include her on most of our projects. Caitlin is helping us produce engagement that draws directly from data that is produced during our grants and creates education modules that are then freely shared. We're actively putting those on. So there is the biological, chemical, oceanography. What is DEMO stand? BICO-DEMO is the data management tool for biological and chemical oceanography. Data management organization. Anyhow, we're working with them actively to create data outreach, visuals, video, data sets that all come from cruises that are then freely available to the public through NSF's data portal. We're working with teacher groups to try to pilot ways that that can be embedded into teaching at community college level as well as high school level. Caitlin also engages actively with the public school systems here, and we have a number of plankton modules. I personally often give modules. I give the plankton talk and then they do a night tow. What's cool about that is that we're starting to create this night tow where we are collecting animals during the day and collecting animals during the night off the dock here at BIOS. We're putting them through that same ZooScan. So we're creating a mini time series of what's going on here. So every group that comes has some ownership over this longer time series, understanding how data works. Some of the more informal things is I work with a historian, philosophy artist who comes out to sea with me, and she wrote her master's degree was on sort of the history of ocean acidification research. She works with us now. I have a collaboration between her and an engineer who does biomechanics, where we're all working on a children's book together about how there can be more than one way to know something and to love something, and that we can all appreciate these organisms. This is specifically the pteropods, how they swim, their ecology and their life, and how each of us from a very different perspective experiences them. And the goal of that project is to, again, provide a window for the younger generation to these things that they never see. I couldn't even spell pteropod until my second year of graduate school, right? How can you care about that? And how can you care about whether your lens is someone who's interested in science or someone who is interested in art and humanities? So there's a bunch of that kind of stuff. The new video game, we're seeing how that's going to play out. And then, yeah, working with a journalist who's writing a book about carbon in the ocean. So those are, I think that covers a lot of it.
That's fantastic. Yeah, there's another question. Do you have future interest to look at carbon flux between deeper zones? For example, meso.
Oh yeah, absolutely. The tricky part is that we're just starting to get the technology rated. So what I mean by rated is like, if you send it too deep, it's going to explode, right? So we're starting it off in the like 1,000 meter zone. And then we'd absolutely like to go deeper. What gets tricky is the deeper you go, the less frequent organisms and particles are. So the more observations you need, but the harder it is to go down there and collect, right? We haven't been focused on doing nets deep here. We have the capacity to do it. Again, for the same reason, is you need to drag the nets through the water a lot longer before you can start saying anything. And this location in particular has really low biomass. And so it's just like kind of tricky to get down there, but oh my gosh, yes, absolutely. We'd love to do deeper things and to get to that point of sequestration, because the easiest way for something to really reach the benthos is in the form of a particle, right? And so zooplankton and that repackaging piece actually gives carbon the greatest opportunity to reach the bottom. So understanding that particle pool business here, where we don't have a lot of phytoplankton based particles, aggregates, it's really critical. We just haven't gotten there yet.
I have a question. Amy, when we began, you were talking about the uniqueness of where you are, in Bermuda and the Sargasso Sea. And I just wondered if you could kind of bring that to the audience, because it really struck.
Oh, I don't even remember what I said. We have just this amazing access to the ocean, right? So before I was here, I'd go on these like big cruises. So I went on this exports cruise to the North Atlantic, and you take like a week of steaming to get where you are. You spend 4 weeks there, and then you take a week to get back. Here, we can take a small boat out for like 40 minutes, throw some nets in the water, collect some samples, and then we can bring them back to the lab, right? The lab where you sleep in a nice bed, people don't throw up everywhere. And you can use all sorts of different experimental procedures that you can't do. So all of this biomechanic work that I was doing, this is like a guy shining like 3 lasers at a swimming organism. And you can't do that on a ship to look at particle speeds. It allows you to go out more frequently to get these finer resolution things and to test things with a little bit less risk. So that part's amazing. It's also one of the most biodiverse places globally in the ocean system. So we're kind of at this, we think of ourselves as like subtropical, but we're actually right next to the gyre, but every once in a while we get this Gulf Stream or mesoscale feature eddy moving through. Sorry, I got very science there for a second. And you get a whole bunch of different animals that come by. And so there's this shuffling. We're at the edge of this. And what's interesting about that is if this place becomes more tropicalized as climate change occurs, we know that the size distribution of organisms in warmer water skews towards tinier things. And tinier things have faster metabolisms, but they also migrate less. So if we move from a community that has big migrators to a community that's small with less migrators, making that calculation of how that's going to shift the ecosystem and the movement is like really interesting because we're at that edge. We already see some of that seasonally, which lets us think forward. Is that what I was talking to you about, Mickey? I don't remember.
Yes, and I was mesmerized by the zooplankton, which I think now from listening comes from maybe one of the scans, the picture, and you were just saying how sort of it was, you know, one scoop and
oh yeah, absolutely. Oh, and don't get Leo started on how many like new species we probably have in our freezer that we just don't have time to do anything with. Like really, truly, this is a place where it's like, oh, yeah, never seen that one before.
That's fantastic. We had one question about whether there would be a recording made from today's talk. And yes, there will, and we will be sharing that as well. So
not to students. I need students.
So I don't see any more questions coming in, and we're nearing we're 1 minute anyway. So I'd like to take this time to say thank you very much, Amy, we will have the recording made for everyone, and we appreciate your talk very much. Thank you.
Thanks, guys.
Bye, everyone.